Welcome to Microlab Mastery. Today's topic, Coagulase Test. The Coagulase Test is a differential test that allows us to differentiate Staphylococcus aureus from S. epidermis and other gram-positive cocci. S. aureus is resistant to normal immune response and antimicrobial agents partly due to the production of coagulase enzyme. So, the coagulase test allows us to distinguish pathogenic strains from non-pathogenic strains. During this test, the coagulase enzyme works in conjunction with normal plasma components to form clumping or clots. The coagulase enzyme occurs in two forms, free coagulase and bound coagulase. There are two different procedures, the slide test and the tube test. The slide test can detect bound coagulase, is simple, and you've had the results within a minute, but it can give false negatives. The slide test demonstrates clumping of cells. The test tube test detects bound and free coagulase, is definitive, but it can take 24 hours, and it detects clots. The materials you need to perform the coagulase test include bacteria cultures, microscope slides, rabbit plasma, sterile transfer pipettes, sterile saline, and materials for aseptic technique, for example, inoculating loop and Bunsen burner. First, label your slides using a Sharpie. Next, place a drop of sterile saline on the left-hand side of each slide and a drop of rabbit plasma on the right-hand side of each slide. Next, please follow aseptic technique to sterilize your inoculating loop, as you have previously learned in the class and as is being demonstrated here. Next, please transfer a loopful of one bacteria to 1/2 of one slide, making sure to thoroughly mix the bacteria into the solution. Then, transfer another loopful of that same bacteria to the other half of the same slide, making sure to thoroughly mix the bacteria into the solution. Please repeat this step 3 using the other slide for the other bacteria. Next, please observe for clumping within 2 minutes. Clumping after 2 minutes is not a positive result. In these examples, slide B with the plasma is a positive result, demonstrated on the lower right of this image, whereas slide B with the saline, slide A with the saline, and slide A with the plasma are negative results. Finally, please properly dispose of your slides and the bacteria cultures into the hazardous waste bag. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.